So in the last lesson, we talked about embryonic stem cells, and we saw that they had the ability to become every single cell type in our body. Now we're going to look at how stem cells can be used in medicine. Okay, I'm going to start a little schematic that I have here on PowerPoint. Okay, and it says cell division is tightly controlled. Okay, so by cell division, I mean when cells divide in two. So when cells grow, they divide themselves in two. So one cell becomes two cells. And then those two cells can divide themselves in two. And then you end up with four cells. Okay, so that's what I mean by cell division. And then cell death is when cells die. And I said here cell division is tightly controlled. And I've drawn a kind of a seesaw. Okay, and that tells me that it needs to be balanced. These two processes needs to be balanced. If lots of cells are dividing, then lots of cells also need to be dying so that you never end up with too many cells. Okay? If you do have cells dividing more than they're dying, that can lead to cancer. Okay? So cancer is when you've got cells that are growing really, really quickly. And they're growing so quickly that they end up forming big lumps of cells called tumors. So when cells are dividing quicker than they're dying, you can end up with cancer. That's what cancer is, when your cells are just growing too fast and you're ending up with big lumps of cells called tumors, which can stop organs working in the correct way. On the flip side, if you've got cells dying quicker than they're growing, you can end up with all sorts of different diseases. And I've mentioned just two of them here below, Parkinson's disease and Alzheimer's disease. So both of these are diseases that affect your brain. So in Parkinson's disease, you've got certain cells in your brain that start dying and they're not replaced. And Alzheimer's disease is the same. You've got cells in your brain that are dying and that aren't being replaced. And because these diseases are affecting your brain, there's all sorts of horrible side effects. So Parkinson's disease, you start off with not being able to control your movements. So everything is controlled from your brain. So when these certain cells begin to die in Parkinson's disease, you lose control of your movements, and you'll start off just with a tremor, but then eventually that will become worse and worse. And then Alzheimer's disease kind of affects all sorts of different areas in your brain, and it can affect your memories, it can affect your speech, it can affect your movements as well. So you've got cells dying that aren't being replaced. It can lead to all sorts of horrible diseases. So before we move on to look at stem cells, we're just going to jot down these two little schematics in our books, okay? And we're just gonna say cell division is tightly controlled. We're not going to go into the control mechanisms, okay? Just take my word for it that cell division is tightly controlled. I'm just gonna draw out my little seesaw, because I think it's quite easy to draw. Okay, and I've got cell division, and I've got cell death. And in this case, they are balanced. Okay, so you want your cells to grow, but your cells should also die. Your cells should be growing to replace the cells that have died. You don't want them growing if no cells are dying, and then similarly, you don't want the cells dying if no new cells are going to grow and replace them. Okay, so you need this process slightly controlled. You want it to be balanced. If the scales tips, okay, so if it tips towards cell division, that can lead to cancer. Okay, if cells are dividing quicker than they're dying, that can lead to cancer. And then if the balance tips the other way towards cell death, that can lead to, I'll say Alzheimer's, okay, and Parkinson's, and there's quite a few others. Okay, so when we looked at health and disease before, we focused purely on, mostly on communicable diseases, and we looked at diseases that were caused by viruses and that were caused by bacteria. But in these lessons, we're going to focus on diseases that are caused by cells dying quicker than they should, okay? And there's lots of different reasons for cells doing this, okay? But when cells die quicker than they should be and they're not being replaced, we're going to look at these different types of diseases, okay? And we're going to look at some of these diseases in detail, but in today's lesson, we're just going to have a look at how stem cells link to these diseases where cells are dying quicker than they are being replaced. Okay, so I'm going to show you again a picture here called stem cell therapy. I'm going to zoom in a little bit. Okay, so we covered the start of this diagram in the last lesson. Okay, so we saw the sperm met the egg. You got a fertilized egg. That fertilized egg grew until it was a big ball of cells called an embryo. And within that embryo, you have embryonic stem cells. Okay, now if this is just happening normally inside the female body, this embryo here slots itself into the uterus, and it stays in the uterus, and it grows and develops into a fetus and then into a baby, and then that baby is born. Okay, but in stem cell therapy, what happens is that instead of allowing that embryo to develop into a fetus and then into a baby, at this point here, a few days after the sperm has met the egg, those embryonic stem cells are taken out of the embryo. And those embryonic stem cells are then grown in a dish. Okay, they're grown in a dish in the lab. And the reason they're grown in a dish in the lab is that these embryonic stem cells have the ability to become every single cell in the body. So if you're growing embryonic stem cells in the lab, then you can turn those stem cells into any other cell type. So for example, if you had someone with Parkinson's disease and some of their nerve cells were dying in their brain, then you've got your embryonic stem cells. You give those stem cells certain signals. That will turn those into neurons. And then you've got these nerve cells that you could transplant back into a patient with Parkinson's disease. Okay? You could have a patient with diabetes. And in diabetes, it's your pancreas that is affected. So you've got your embryonic stem cells. So you take them from an embryo, you're growing your embryonic stem cells. Then you give your embryonic stem cells certain signals, and then they turn into the pancreatic cells, which can then be transplanted back into the patient. Okay, and this is called stem cell therapy. You start with the embryonic stem cells. You give them certain signals to turn them into the specialized cell type that you want, and then you've got all these cells that you can transplant back into the patient. Now, there's all sorts of obstacles to overcome with this. You might be able to spot a few yourself, but that is the general principle of stem cell therapy. Okay, before we go any further, I want to make something clear. These embryos are not being taken from the uterus of a pregnant woman. Okay, these are embryos that would have been made in the lab anyway. So hopefully you've heard of IVF, so in vitro fertilization. So you've got couples that might struggle to get pregnant. And what will happen is that the female will donate her eggs and the male will donate sperm. And then this process will all happen in a dish. So the sperm and the egg will meet in a dish and you'll form fertilized embryos. Okay. And because the woman will usually donate a couple of eggs, the man will donate lots of sperm, you could have like 10 or 20 fertilized eggs. You're not going to implant all of those eggs back into the mother. Okay, because you don't want to risk her having multiple babies at the same time. So typically you'll insert 1 or 2 fertilized egg back into the mother. And then you've got all these other fertilized eggs. And it's totally the parent's choice what to do with them. They could freeze these fertilized eggs for forever. You know, they never have to do anything else with them. Or they could decide that they've had all of the children that they want to have and then donate those fertilized eggs to science. So they're their eggs, and they can use them as they see fit. They could have multiple pregnancies, a few years spread apart. And then after using 5 or 6 of their fertilized eggs and they've had a few children and they don't want any more, and they've still got 10 of their fertilized eggs sitting in a freezer, they could then donate them to science. So these are all fertilized eggs that have been donated to science. They are not being removed from a woman's uterus. Okay, so this is all done in a lab. And any fertilized eggs that are donated to science, you would take the embryo, you can remove the stem cells from the embryo, then you would grow the stem cells in a dish, and then, as I say, apply different signals, which is obviously slightly more complicated than what I'm making it out to be, but you can apply different signals to these embryonic stem cells to turn them into the cell type that you're interested in, and then transplant them back into your patient. So we're going to jot down just a few notes on that so we've got them in our book. Okay. So I'm going to write under here, stem cells can be used to treat diseases where, I'm going to say, damaged or dead cells need to be replaced. An example of that would be Alzheimer's disease, where you've got cells, nerve cells in your brain that are being damaged and that are not being repaired. Okay, so stem cells can be used to treat diseases where these damaged or dead cells need to be replaced. Okay, I'm just going to write, this is typically done with embryonic stem cells are harvested from embryos that have been donated. Okay, they're not being removed. They're harvested from embryos that have been donated. The stem cells are grown in the lab and converted into the cell of interest. Cell of interest, e.g., nerve cells in Alzheimer's disease. Okay, so that's the general gist of what would happen. Okay, you harvest these stem cells, you grow them in a dish, you convert them into the cell you're interested in, and then you try and transplant them back into the patient to replace the cells that have been damaged. Okay, and at the moment I've just mentioned diseases, I've mentioned Alzheimer's, I've mentioned Parkinson's. This could equally work if someone's been in an accident. Okay, so if you've suffered a spinal cord injury in an accident and you find yourself paralyzed, the idea is that you could grow new cells from stem cells and then transplant them into the patient who is paralyzed to replace the cells that have been damaged. Okay, so there's lots of potentially, and I keep saying the word potentially, different uses for stem cells in medicine. Before we look into the uses of stem cells in medicine a little bit closer, there are obviously ethical issues surrounding this. I'm just going to show you a cartoon that kind of sums this up. Okay, and the main ethical issues surrounding the use of embryonic stem cells was that, in order to harvest the embryonic stem cells, you are destroying an embryo. And this cartoon, well, I'll let you have a look at it for yourself. Or you can pause if you want to have a proper look at it before I start talking about it. But what you've got here is you've got a depiction of heaven. Okay, you've got this kind of older gentleman here who said he died waiting for embryonic stem cell research to find a cure. Okay, so there's so many different diseases that just don't have cures, and so many really, really debilitating diseases that just don't have cures. And this gentleman has obviously suffered from one of those. He's died for a disease that they don't have a cure for. And then here you've got someone else who said, I was the embryo. So he's waiting for embryonic stem cell research to find a cure. And this little chap here was that embryo who was destroyed in order to find the cure. So with these cartoons, they tend to favour one side over the other. I just want you to think to yourself for a second, which side of the debate do you think this cartoon is on? Do you think this cartoon is pro-embryonic stem cell research? Or do you think this cartoon is anti-embryonic stem cell research? Just pause me for a second, pause the video and think about that for yourself first before I start discussing it. When you're thinking about it, think, is there any kind of look at how they're depicted, and that will give you a good clue as to which side of the debate this cartoon is in favour of. Okay, so I'm gonna start talking about it. I would say that this cartoon is anti-embryonic stem cell research, and the reason I would say that is just how the embryo is depicted. Okay, remember, an embryo is literally just a ball of cells, and when this cartoonist is depicting it, it's showing them as a baby. So, if you destroy an embryo, that embryo is never going to develop into a baby. Okay, but remember where scientists are getting these embryos. Okay, they're being donated by parents who don't want to have any more children. So if they haven't donated these embryos, what's going to end? What are these embryos going to end up doing? I mean, more than likely, they're just going to sit in the freezer, indefinitely. So these are donated by parents who are never going to have these children. So it's either they sit in the freezer or they're used for embryonic stem cell research. However, having said that, each embryo is unique because each sperm is different and each egg is different. So each embryo is going to produce a unique person, just like you're all different from your brothers and sisters. Each embryo does produce a unique person, and by destroying that embryo, you are destroying that life. That life can never be fulfilled once you destroy the embryo. So it is controversial. There is ethical issues around it, but the way the embryo is being depicted here as a baby is very incorrect, and it's kind of trying to tug at your heartstrings and make you see that a baby's life is being destroyed. In reality, you need to remember that an embryo is just a ball of cells. Only 5 or 6 days after the sperm has met the egg, it does not have any facial features or look anything like a human at this point. But yeah, what the cartoonist is trying to get across is that this chap here died anyway. Stem cell research didn't cure his disease, he still died. And not only has this guy died, but you've also robbed the life of this embryo in the process. So there's lots of ethical issues surrounding the use of stem cells, and one of them is just being highlighted here in this cartoon. I'm going to give you a few of these questions to do. The first 4 I think should be okay. The 5th one we haven't discussed yet. So I've just spoken about embryonic stem cells being used in medicine. I haven't mentioned adult stem cells at all in this lesson, but I've asked you to suggest a reason why embryonic might be more useful than adult. So have a think about that one for yourself. The other 4 you should find the answers to in your notes from this lesson and the previous lesson, but 5 you'll have to think about for yourself. So press pause and answer those questions for yourself, and then press play again. Okay, so I've included the answers to the questions. So how do you get embryonic stem cells? You harvest them from embryos that are donated after IVF. So IVF is just in vitro fertilization, and that's fertilization, so the sperm meeting the egg in a dish, okay? And that's occurring in a lab because couples are having trouble getting pregnant. So the man donates the sperm, the woman donates her eggs, and that fertilization process is happening in a dish, okay? And any embryos that are unwanted, you can harvest embryonic stem cells for. Ethical issues, you are destroying an embryo. And if you're destroying an embryo, you are preventing that embryo from ever developing into a person, okay? Some people would say if you're destroying an embryo, you're destroying a life, but it depends on when you define life. Some would define life as when the baby is born. Some would define life as, a certain stage of pregnancy, like 20 weeks or 30 weeks. Some would define life as the second the sperm meets the egg. So it kind of depends on when you would define life, which is, again, why it's a contentious issue. It's the same with abortion, where people would strongly feel that, even after a couple of weeks, that it's a life and you've got no right to destroy it. It's the same with an embryo. Although it's just a ball of cells, some would argue that it is still life and you've got no right to destroy it. So I've written destroying an embryo, and so preventing that embryo from developing into a person, as opposed to just saying destroying life. It's more grey as to how you define life. 2 locations where adult stem cells are found. So I wrote the intestine, which is in your digestive system, the skin. You could also write bone marrow. That's where blood stem cells are made. You can say, there's a couple of others. You can say breasts, testicles, brain, surface of the eye. There's a couple, okay? But the reason they're needed is for growth and repair, okay? So not all of the cells that exist in your body have the ability to grow. So you've got this pool of stem cells that do have the ability to grow. So for example, your typical blood cells, your red blood cells and your white blood cells, they themselves can't grow. So you've got these blood stem cells that replace them when they get damaged, okay? The same in your skin, okay? The skin cells themselves don't grow, but you've got these skin stem cells that replace them as they get damaged, okay? Why are embryonic stem cells more useful? And this is something we're going to talk about a little bit more in later lessons when we look at specific diseases and how they can be treated. But number one advantage is that embryonic stem cells can become every cell type in the body. So remember, I told you with adult stem cells, so if you've got a brain stem cell, it can only become brain cells. If you've got a skin stem cell, it can only become skin cells. But if you've got an embryonic stem cell, it can become every single cell type in the body, which just gives you way more potential. They're also much easier to obtain. Okay, if you've got your little embryo, I'm not going to switch back. If you have your little embryo, there's only a few cells in that embryo, and a lot of those cells are embryonic stem cells, and it's really easy to get them out and to separate them from the other cells. If you've got like your brain and you're trying to isolate the few brain stem cells amongst all of your other brain cells, it's really, really difficult to isolate a pure population of stem cells from a whole organ. So it's much, much easier to get those stem cells from just your embryo. And they're also younger and healthier. So when you've got an embryo at the very, very start of life, those cells have the potential to grow so quickly. Okay, your adult cells, they just don't grow that fast because they don't need to. So these younger, healthier embryonic stem cells, they're going to grow a lot faster than adult stem cells will. And you do need to be able to grow them up in the lab. So embryonic stem cells have huge advantages over using adult stem cells, which is why they are typically preferred despite the ethical issues that accompany them. Okay. So for homework, there will be a quiz on Show My Homework this week that just kind of tests you on some of the knowledge that we've covered in these two lessons. And in the next couple of lessons, we're going to look at individual diseases in more detail to actually see, are stem cells being used to treat them? And if so, are they effective? And if they're not effective, what are the problems with using stem cells? Why are they so challenging to use?